Hi, good evening, and welcome to our Reef Relief annual membership meeting. Thank you all so much for joining us tonight, and thank you for all of your support throughout the past year, through your donations, attending our fundraising events, volunteering, or trusting us with your kids in Coral Camp. Your support has been more important than ever this year. 2021 continued to present unique challenges, but with your support, we've been able to adapt and overcome these challenges, even expanding our education programs. Through our virtual programs, we're now able to reach students, not just in our community, but throughout the country and even around the world. We've been able to return to in-person volunteer events, continuing our outreach within our local community. And of course, we continue to fight for our coral reefs through our partnerships with advocacy groups, such as the Everglades Coalition. We have some great speakers for you tonight, who I'll introduce in a few minutes. But first, I'd like to introduce myself. My name's Tricia Coyne, and I'm so honored to introduce myself to you, the membership, for the first time as the president of your board of directors. Joining me tonight is our vice president, Carly Chabot, our secretary, Mimi Stafford, and our treasurer, Genya Yerkes, as well as our other board members, Bob Cardenas, Maggie Howes, Ed Russo, Kevin Keeley, Dr. Patrick Rice, and Alice Reeses. As well as our staff tonight, program coordinator, Taylor Sukumis, education coordinator, Erin O'Brien, our moderator tonight, education director, Dora De Maria, and of course, our incredible executive director, Mill McCleary, who I'd now like to give the floor to tell you a little bit more about the work that we've been doing in the last year. Mill?
Sure, thanks, Tricia. Yeah, I'll keep it brief because we've got such great presenters for this evening. I want to start by dittoing what you said and thanking our support. It's been an incredible, it's been another challenging year, but we've had a lot of accomplishments, mostly due to our partnerships with the nonprofit community, government partners, all of the interns that gave their time to us throughout the year, the business community through either hosting fundraising events or being a dues paying member itself, or donating the many in-kind donations to help keep our programs going. I want to give a shout out to the awesome foundations that have backed our work tirelessly through the years, and especially over the past year, but most of all our members. And those of you that are attending tonight that are dues paying members, thank you so much. And to those that can't join us tonight, we couldn't accomplish what we do without your support. Our finances are as healthy as they've ever been because of the support of our memberships and foundations and communities and others. But like I said, our finances are in great shape. And because of that, we've been able to expand. Tricia mentioned our new staff members. Taylor and Erin have been doing an extraordinary job. They've both started within the last year. Dora's been promoted to the education director, which has been fantastic for us. And Alex, although she left our staff, she's now been appointed as a board member. So she's sticking with. So we're seeing growth, and in part, or much in part, to our healthy finances. Like I said, it's been a challenging year, but things are slowly getting back to normal. I'm glad to report that our environmental center, which you may have seen in one of the slides at the beginning before the meeting started, it's now reopened to the public. You can find us there at 631 Green Street, Monday through Friday from 10 to 4. So we're happy to be reopened. And we've already seen an influx of visitors looking at our exhibits and getting information on how to join and purchasing our retail t-shirts and hats and stickers and things like that. So that's been great that the center's been reopened. We're finally and very happy to report that we've been able to do things in person with our volunteers. Over the last year, we've worked with 431 volunteers through our stormwater education programs, such as stenciling storm drains and marine debris removal, to give you a couple examples. And over the last year, with those volunteers, we've removed 6,480 pounds of marine debris. So we see that growing in the coming year as things slowly get back to normal and we're able to do things more and more in person. Including our Big Kids Coral Camp, that came back this past summer in partnership with the Dry Tortugas National Park. We were able to take our older campers out camping for 3 nights in the Dry Tortugas. And so that's been great that's been back up and running. In fact, our education programs have grown over the last year. We were limited over the last couple of years with the amount of coral campers we were able to have in order to safely social distance and such. But we've grown to 214 campers over the last year. We expect that number to get back to normal this year, to be above 250 campers. Our Discover Coral Reef School program, we're able to now visit schools in person once again. And already in the last year, we've seen over 6,000 students at over 20 schools between Key West and Fort Lauderdale. And as Tricia mentioned, our virtual learning platform has reached over 205,000 adults and students, not just locally, but around the world and nationally. So that's been fantastic. And you can learn all about those programs and see how to sign up for them by going to reefrelief.org, or you can email us if you have questions about that at reefrelief@gmail.com. And while we are continuing to grow in that respect with our education programs and our partnerships, we also are continuing to work to improve water quality, not just with our stormwater projects that we do locally, primarily in the lower Keys, but we continue to, as a member of the Everglades Coalition. And in fact, they are having their first conference back in person in a few years in January, and it's going to be in the Keys at Hawks Cay Resort. And the registration is now open, and you want more information about that, you can find that. You Google the Everglades Coalition Annual Conference, you can see the links to register for that conference. It's from January 6th through 8th. We've had good and bad news with our sunscreen campaign. I think most of you know that we became the first place outside of Hawaii to ban harmful chemicals from sunscreens to corals. But unfortunately, the state preempted that and isn't allowing us to enforce the ban. But in turn, we worked with the City Commission to pass a second resolution, an ordinance to do a deeper consumer education program. So we're not giving up in that effort. And in fact, there's going to be some bills introduced in Tallahassee this next session that we're going to be looking at closely that will help to either reinstate our ordinance here in Key West, but also to expand it to the entire state of Florida. But with that said, with this second ordinance that the city passed, we filmed just as one example of our continued sunscreen education, we filmed a 60-second promo that is being aired multiple times daily by Keys TV. So visitors to all the Florida Keys are seeing that in their hotel rooms every day, multiple times a day. So we're continuing with that education effort. That promo runs through the end of May 20 of next year, and we hope to obtain some more foundation money to continue that into the future. And we continue to grow our Skip the Straw campaign up the Keys. And while we're doing that, we're still putting in efforts to grow in our clean water campaign efforts. So you'll see some new ideas and ways about doing that in the coming year. And in closing, on a couple fun notes, some of you know that the Key West Monopoly has been released, and we were the organization that benefited from that. And you can find your game, you can purchase the game at the Concord Public Seafood Company right next to Reef Relief. So you can come on down there, purchase the game, support Reef Relief, and then stop on by and visit us. And then also we, over the last year, we've been working with the Grateful Dead family, and we've been able to trademark the Grateful Diver line and helped launch the gratefuldiver.com website, where you can purchase apparel, stickers, hats, and such to support our program. So just to close on a couple fun notes there, the Grateful Diver line and the Key West Monopoly, which is kind of a fun thing we did in the last year. And with that, that's what I got for our report. So again, thank you so much to our membership and our staff and our board of directors and all of our supporters that we've had over the last year. We really appreciate it.
Well, Mill, thank you so much for all of your amazing work. It's really, really impressive. As I said before, we have some wonderful speakers tonight. We will have some time for questions after each speaker, which will be moderated by Dora. So if you have any questions, please send them to her through the chat function, not the Q&A function. If you can send it to her via chat, and then she'll make sure that your questions get asked after each speaker finishes. So our first speaker tonight is Dr. Karen Neely. Dr. Neely is a research scientist at Nova Southeastern University, who's currently researching the impacts of and responses to stony coral tissue loss disease. She conducted initial laboratory treatment trials, pioneered field intervention efforts in the Florida Keys, and with her team has treated nearly 10,000 diseased corals in Florida. She also documented the rapid decline of the iconic pillar coral population as a result of disease, and she coordinated the rescue of over 500 fragments of the species, which are growing and producing the next generation of corals at partnering facilities. Dr. Neely has lived and worked in the Lower Keys since 2001, conducted over 3,000 dives, and has authored over 20 marine science publications. Her presentation to us tonight is Corals in Crisis, Responding to Stony Coral Tissue Loss Disease.
Great, thanks, Tricia. Can you guys hear and see me okay?
Yes.
Ready?
Yes.
Okay, thanks. All right, let me pull up my presentation here and we'll get started. Okay, I just want to confirm that you can see a title slide, and I can't see anyone, so if someone could give me a verbal confirmation, that'd be great.
Looks good.
Great, thank you so much. All right, good evening, everyone. Thank you so much for being here. Thanks for your interest in reefs and for your support of Reef Relief and the work that has been happening here in Florida. I want to thank also Reef Relief for extending an invitation for me to share our work, and to Dora in particular for coordinating all of this. Much appreciated. So I'm Karen Neely. I'm a research scientist at Nova Southeastern University. Nova is based out of the Fort Lauderdale and Dania Beach area, but myself and my team live in the Florida Keys, everywhere from Key West up through Marathon. I first came to the Florida Keys in 2001 as an instructor with Sea Camp. I fell so in love with the reefs that I turned that into my graduate work, doing work on Florida's corals, and then turned that into a career. Many years at FWC, some work with what was then Florida Keys Community College, and now with Nova Southeastern University. So I'm really delighted to share the work that's been happening by myself and my colleagues really over the last decade. So I want to just start with some acknowledgements. All of this work has been permitted under a huge number of permits from our national parks, from Florida Keys National Marine Sanctuary, a huge variety of funding sources, including the state, DEP and FWC, some federal funding from the EPA, from NOAA's Coral Reef Conservation Program, and from NOAA National Fish and Wildlife Foundation Coral Emergency Response Fund. This all would also not have been possible without huge contributions from all of our collaborators and partners who've donated their time, their resources, their funding to really make this happen. This has been a huge undertaking, so appreciation to all of them. I'm sure that anyone attending the Reef Relief annual meeting already thinks that reefs are pretty awesome and has a pretty good idea of why they matter. So I'll just hit on a couple of high points here. The first being the biodiversity. Reefs don't cover a lot of the Earth's surface, less than 1%, but they make up more than, or they contain more than 1/4 of all marine species. In terms of fishing and food and reef's contribution to that, about 50% of US federally managed fisheries depend on coral reefs at some point in their life cycle. So if you like fish, you probably like coral reefs. Certainly here in South Florida and Monroe County in particular, tourism is such a backbone of our economy, and coral reefs are a huge part of what draws people to our area. Some of the hard numbers here, this is from a 2013 NOAA report looking at Monroe County and the South Florida region. This is the amount of jobs, the amount of employment income, and the amount of sales that are attributed directly to the presence of coral reefs. And you can see that those numbers get into the billions of dollars. So economically, a huge driver. Something we tend to forget about until we're standing knee deep in saltwater in our living rooms is how important coral reefs are to coastal protection. Some really interesting work just came out of USGS this year. I would encourage you to take a look at this report. It's a evaluation of what coral degradation will have to our coastal hazard risks and the economics associated with that. So their data is publicly available. I was able to pull that and make a map of the Key West Stock Island area. And this yellow is basically the areas you would expect to flood on a 10-year storm event, so kind of a 10-year interval hurricane, and under our current status of coral reefs. Now, coral reefs are projected to continue to decline without active intervention, and they've estimated that with a decline of really only about a foot, so really that top veneer of coral disappearing, this is what that same storm would look like. I'm sure there's some people on this call who live in what is now that orange area. And so that's the difference that that little top foot of coral reef makes in terms of protecting our island. What does that look like in terms of the money side of things and the people side of things? So this is basically the annual loss of coastal flooding protection as a result of coral reef degradation. So the loss of that foot or so of coral annually, this is the amount of people, the value of the buildings, and the economic activity valued at risk because of that loss. And again, we're talking hundreds of millions of dollars here. So these reefs are providing us with invaluable services, and we do really very little to pay them back for that. So the decline of reefs is not new and probably not a surprise to anybody on this call. We know that coral cover has been declining, not just in Florida, but throughout the Caribbean for decades. You can see on the right here some photos of Carey's Fort Reef taken a couple decades apart. Those of you that have been around have seen firsthand this decline. And unfortunately, this is not unique to just Florida either. The cause of this, though, is a little bit more difficult, and we often talk about the death of reefs by 1,000 cuts. And that really presents a lot of challenges. If there's a single cause to a decline of an ecosystem, it's a lot easier to try to find a solution to that problem. But when there's multiple problems, it can be a lot harder to focus your efforts onto specific solutions and a lot easier for each problem to shift blame to some other problem. But some of the ones that we really can't ignore in the room: warming temperatures, declining water quality, and we know that all of those stressors can contribute to things like coral bleaching events and also to diseases. And all of these cuts to this reef, these 10,000 cuts, really range from something very localized, like a single anchor dropping onto a single coral, or a single piece of marine debris that might be smothering that coral and can be picked up by a diver, to really much more global problems like climate change that also have these impacts. So we're dealing with a lot of problems that are going to require a lot of solutions. Now, I talk about this death by 1,000 cuts. But there's a new cut in the room, and it's a really deep one. And so a lot of the responses that I'm going to talk about tonight are in response to this new threat called stony coral tissue loss disease, or SCTLD. And this was a disease that appeared in 2014 off the coast of Miami. It was very quickly determined to affect about half of Florida's coral species, and that includes most of the reef builders, so most of those big boulder corals, the brain corals. A notable exception is the branching corals, so the elkhorn and the staghorn, which have been the species that have been most outplanted by different restoration teams, have largely been spared the brunt of this disease. But for really most of the other corals on the reef, this is a game changer in terms of what their populations look like and what their futures look like. Across the bottom here, I've pulled some quotes from various peer-reviewed papers. Scientists are not particularly prone to hyperbole, but you can see the extent to which this disaster is sort of unfolding even in the peer-reviewed literature. So a couple of rather unique characteristics of stony coral tissue loss disease. the rate at which it kills corals is very rapid. So within weeks to months, huge corals that might be hundreds or thousands of years old can be completely wiped out. There's also a really high infection and mortality rate. So depending on the species, between about 66 and 100% of individuals will contract and die as a result of this disease. So extremely high mortality rates. And then one of the bigger issues is just how unprecedented this disease is in the geographic and temporal scale. And what that means is that disease is a part of any ecosystem. It's there. There's what we call background rates of disease. This happens in human populations. This happens on the reef and in forests. But when you get these outbreaks, most of the time a disease outbreak will be pretty short-lived in terms of its geographic span and also its temporal span, how long it lasts. And then it generally fades back into the background. What's unique about SCTLD is that even 6 years, 2014 to 2021, 7 years after its arrival off of the coast of Miami, corals there are still contracting this disease and still dying from this disease. And it didn't just flare up near Miami and then simmer back down. It has unfortunately spread really quite widely. And all of these characteristics have really led to this being what we're considering unprecedented, devastating, catastrophic, all of these words describing what we're seeing on our coral reefs right now, even with this background of 1,000 cuts that we've been dealing with for decades. So this is a figure that Florida Department of Environmental Protection put together. This is where the disease was first documented in 2014. The prevailing currents off of Miami is the Gulf Stream pushing things north, and sure enough, the disease sort of followed suit really quite rapidly, making it up to the northern part of Florida's coral reef. It also progressed more slowly, but really quite steadily south, progressing through the upper Keys, middle Keys, lower Keys, and unfortunately, as of the end of May of 2021, reaching Dry Tortugas National Park, and it continues to spread throughout the national park there. This is an example of what a reef that's in the throes of stony coral tissue loss disease outbreak looks like. This is off of Long Key, and you can see a wide variety of corals here. There's brain corals, there's some boulder corals, a diversity and a relatively high coral cover. But anywhere that you see that bright white, that's a coral that is actively dying or has very recently died. And to give you a sense of the ones that aren't going to make it versus the ones that are okay in this photo anyway, all of those red dots represent corals that were alive probably 2 months before this photo was taken, and all of which were probably dead within about a week or two after this photo being taken. So this is a huge loss to the diversity and to the coral cover, all of which contribute to those ecosystem services that we talked about earlier. I unfortunately had a very front row seat to the impacts of stony coral tissue loss disease on our Florida population. And that was because of my long-term work with pillar coral. Pillar coral, if you've never seen one, is about as cute as corals can get. Most people don't get excited about fuzzy looking rocks, but pillar corals have their tentacles out during the day, so they're fluffy. They've got these big columns that come off of them. We sometimes refer to them as unicorns. Also refer to them that way because they are rare but conspicuous. It's very obvious when you see one, but they have never been particularly common on Florida's coral reef. And so back in 2013, when they were being considered for listing under the U.S. Endangered Species Act, there was some funding and a project available to try to assess this population, really put some numbers to it. How many are there out there? How are they doing? What sort of stressors are they experiencing? And so we started mapping all of the pillar corals that we knew about, and we got information from dive shops and monitoring programs, from restoration groups and just citizens and fishermen who knew about these. And we're able to go out and find about 800 pillar coral colonies throughout the state of Florida. And we were able to track those through time. And so this is what pillar coral looked like back in 2013, 2014. Everything north of Biscayne, I should note, was mapped by Listina Cabay, who was a graduate student at Nova under Dr. Dave Gilliam. And so this represents some of her data as well. But you can see for the most part, these corals were out there, they were alive. There's some background, background disease. The yellow corals are indicating another disease called white plague, but it wasn't particularly prevalent, and these corals seem to be doing kind of okay. In 2015, you see that red arrow there, that's pointing to a dead pillar coral, which didn't seem to succumb to a normal disease. And this was really one of our first indications of something different out there. By the time 2016 rolled around, you can see that this has spread dramatically. And by this point, stony coral tissue loss disease had been named and identified. And all of those red dots represent corals actively infected, the black dots representing corals that have died, almost always as a result of stony coral tissue loss disease. And this really gave us a sad but very accurate timeline of how stony coral tissue loss disease was spreading through the Florida Keys. So by 2017, it was off of Long Key. And so the red is indicating the front spread of it. And then the black obviously follows behind as these corals die as a result. 2018 down into the lower Keys. By 2019, all the way out into the Marquesas. By 2020, almost all of the population is dead. This has continued through 2021 with, unfortunately, all of the colonies out in Dry Tortugas now affected as well. So on the left here, this is what that looks like. The top photo, this was one of my favorite sites. It was called Pillar Coral Forest. There were over 100 colonies at this location. It was always teeming with fish and all sorts of diversity and life. You can see by 2016, after stony coral tissue loss disease has wiped through, it's basically just a pile of standing up columns which have continued to crumble just into a pile of rubble with the associated lack of fish life. And so between 2013 and 2020, we lost about 93% of these corals. And again, that is continuing today. Just to put that in perspective, imagine if we lost 93% of the alligators in the Everglades in 5 years, or 93% of the palm trees in the Florida Keys or the mangroves. This is really the scale we're talking about in this local extirpation as much. So we don't really think about it. So in that sort of worst case scenario, what we were really hoping for was that this would remain a Florida problem. And for about 3 years it did. This was never spotted outside of Florida. And a lot of people were keeping an eye out. This has been so devastating to our reefs that everybody else was very keenly aware of it and wanted to realize if it showed up on their back doors as well. Unfortunately, that was only a 3-year short-lived hope. And by 2018, we started getting reports from Jamaica and from Mexico. And since then, it has continued to spread throughout much of the Northern Caribbean and down the Caribbean arc, including our American colleagues in Puerto Rico and the US Virgin Islands, who are currently dealing with pretty substantial outbreaks as well. I want to point out that while the disease appeared to travel according to water currents within Florida, these distribution patterns throughout the Caribbean really don't follow established water currents. And so there's a lot of consideration as to what might be driving some of the appearances of disease in these far-flung places. And one of the theories definitely revolves around shipping, boat shipping, moving large ships from place to place, especially with ballast water. And if that's a topic that interests you, I really encourage you to check out the Rosenaw 2021 paper that really outlines that argument quite well. All right, so this has been a problem in Florida now since 2014, and it has become a problem throughout the Caribbean. What is the response to this, this catastrophe? And as the extent of the damage grew, and as the number of people involved in any sort of response grew, it became quite clear that there needed to be some sort of organizational structure to deal with this. And so the various governing agencies established what is referred to as the Executive Coordination Team. And this is the leads for the National Park Service, Florida DEP, Florida FWC, and NOAA, who sort of guide this response, looking at these big picture efforts. Over on the right-hand side, you'll see with a dashed line, the Disease Advisory Committee. This is a group of about 200 scientists and managers. We meet bi-weekly to kind of learn the latest. What's going on with this response? What are some new ideas? What are some scientific, technical critiques that might help with some of this response? And then all of the work that was being done got organized into these sub-response teams. And I'm going to focus primarily on these three because they represent the on the ground, in the water efforts to deal with stony coral tissue loss disease. But I certainly don't want to discount these other teams. So epidemiology and research. There are hundreds of people working on researching SCTLD, and we have some substantial knowledge gaps associated with it. The biggest one being that we actually still don't know what causes SCTLD. There's a lot of evidence that points towards a bacterial pathogen, but there's some recent work that came out that also found some viral-like particles in diseased colonies. So really even just figuring out what SCTLD is quite important. Other questions concern environmental conditions that might hasten the spread or worsen the virulence of this disease, and those are all being addressed by this team. There's also obviously some regulatory components, a lot of data being handled, some communications that need to be done in relation to this, and there's teams from these various agencies that are related to that. And then importantly, I think the Caribbean Cooperation Team, because Florida had the unfortunate head start of dealing with this over the rest of the Caribbean, through this team, we've been able to share a lot of the lessons learned, a lot of the mistakes made, and also the successes with our Caribbean colleagues who are then able to kind of get a jump ahead and handle their own responses at a more rapid rate than those of us here in Florida who were sort of battling with this from day one were able to. All right, so these in water actions have really followed two parallel paths that have been working in conjunction with each other. One of these is coral preservation. So there are corals affected by SCTLD on the reef, and those can be saved. And I'll talk a little bit about that, but that has been one of the main focuses of this effort, is to preserve the corals that are still on the reef, the ones that are providing those ecosystem functions. The other is coral rescue and propagation. So this is bringing corals in off of the reef to keep them safe from SCTLD, and also to be able to use them to create new corals. So on the preservation side of things, like I mentioned, we can save corals that are impacted by SCTLD, but there are way more corals than there are of us. There are teams in Dry Tortugas National Park and Biscayne National Park, also some of the counties up in Southeast Florida, along with research collaborators at Nova and at FAU that have been working on this. And here in the Florida Keys National Marine Sanctuary, there's a grand total of 4 of us that are doing this. And so 4 people stretched out over 100 miles of reef. We've had to make some really tough choices as to what corals can be saved, which ones are going to have to be sacrificed. And really those efforts have had to focus on iconic sites. And so that means not just the ones that are sort of famous with tourists, so Lou Key, Sand Key, Case Fort, these places that are sort of common household names with Florida reefs, but also those that really have the most potential to contribute to reefs of the future. So the ones that are providing some of those ecosystem services that have high diversity, that have high coral cover, that are most likely to provide the reproductive output for the next generation. So we focus on those sites. We also have to focus on certain corals within those sites. Again, there's 4 of us and millions of corals out there. And so we generally choose some of the largest. You can see that picture in the top. There's a little diver there. So this coral is about the size of a bedroom. And you can imagine how important that is for all of the fish that live in it and all of the structure that it provides. Also iconic corals, some of the more highly endangered corals get a little bit of extra love as well. So when SCTLD was first recognized as a large problem that wasn't going away, one of the immediate responses was to try to figure out if we could save these corals. And there were a lot of things that were tried as a part of this. No idea was too ridiculous, but we worked with a lot of collaborators, and one of the great early successes was that some of these pillar corals had been sent off to our colleague, Dr. Cheryl Woodley in Charleston, South Carolina. We said, just save them, just do what you can to save them. And she found that if she added antibiotics to their aquarium water, that SCTLD halted and that these corals were fine. And they're still fine. They're growing, they're reproducing, they're great. So this was sort of a sign that there might be a solution here, but we obviously can't just dose the ocean with antibiotics. So we had to get this into a topical form. And again, with some great collaborators, Dr. Cheryl Woodley again, our colleagues at Florida Aquarium, and a company called Ocean Alchemists. They were able to develop a topical paste that we could infuse with amoxicillin and then apply that directly to diseased lesions of corals. So I think about this a lot as just applying Neosporin to a cut on your arm that might have a little infection to it. It's really the same thing. And so with that, we've actually been really delighted with the success of this. 95% of lesions that have this application applied to them halt, and those corals are no longer experiencing mortality as a result of SCTLD. We do go back to each of these corals every 2 months. Every coral gets a checkup. Some corals will get new lesions associated with them, particularly within the first month or two after their first treatment. And so we can provide a secondary care to that. And again, those applications are also effective. And so we're pretty delighted. After 2 years, we've only lost about 4% of these treated corals. And again, if you go back to considering that between 66 and 100% of corals that get SCTLD usually die, we're pretty delighted with that only 4% mortality rate. So I want to sort of show you what this looks like. This is a video that my team member, Sydney Gallagher, took on an intervention trip out to the dry tortugas. So please sit back and enjoy for a minute.
In the nighttime, always in the nighttime, up too late, lying on the floor, thinking I've been here for so long, just a little too long, think I need to get out of the door, but I've never been a brave one, always was a tame one, always double checked all that I knew, but that safety is a danger, yeah, there's only one way out, it's through, through and through, oh, through and through, I'll find my way to you, though I'll admit it may take me some time, oh, through and through. I will be there for you, even if we may seek a separate life. And I don't know what I've been waiting for. It feels like I'm always losing my way. I'm doubting every step I take. I know that everything finds its place, although I wish I knew where to. I guess I'll have to see it through, through and through. I'll find my way to you, though I'll admit it may take me some time. Oh, through and through, oh, through and through, I will be there for you, even if we may seek a separate life. Oh.
So that's pretty much the day in the life of a coral saver. We do get special gifts from the ocean sometimes, like dolphins, or yesterday I had beautiful spotted eagle rays come right up, and that's always much appreciated. But this is the ease, but also the labor intensivity that goes along with this work. So what have we been able to accomplish? I've shown a couple maps here on the top. Again, I mentioned we had focused a lot on some of the iconic reefs of the Florida Keys. And this is the number of corals that have been saved at each of these locations. You'll see up in the upper Keys, those dots represent places like Carysport, Grecian, Molasses. And really by the time we were able to start this work, stony coral tissue loss disease had taken a huge toll up there. There's really just not a lot of surviving corals. But as you see, we get into the inshore sites or into the lower Keys, disease had not progressed very far at that point, and we were really able to get in and have a huge impact at these sites. On the bottom map there, that's a map of Lou Key. This map is about 3/4 of a mile long, and you can see each of those dots there represents a coral that has been saved at Lou. Again, these oftentimes represent some of the biggest and oldest animals in Florida. And you can imagine how different that reef might look with those 1,000 enormous corals dead had we not been able to get in there. One of the ways that we've thought we started to think about quantifying this is something that people are a lot more familiar with, and that's restoration. And restoration is great and super important for restoring the corals that have been lost. What we like to think of what we're doing as is making their lives a little bit easier by preventing more corals from being lost. And so by being able to go out and treat some of these large corals, and you saw how rapidly a coral can be treated, we're really saving the restoration effort from having to go and replant all of those corals. And we've roughly estimated that the surface area that we've saved through our work is equivalent to almost 1 million restoration outplants. And that again provides hundreds of thousands of outplants that can now go towards corals that really died before stony coral tissue loss disease. And so we hope that we're being able to make their burden a little bit easier as a part of this. So as this sort of has been proven in the Florida Keys and translated to other parts of the Caribbean as well, when disease reached Dry Tortugas National Park, we were able to be awarded the very first National Fish and Wildlife Foundation Coral Emergency Response Fund grant. So this was set up as a mechanism to get people out there saving corals after an emergency as quickly as possible, really before it was too late. And this was really a prime opportunity for that. We had a way to save these corals. We knew that this was devastating. And so we're really grateful that NOAA and NIFWF had the confidence in us to be able to take this on. And sure enough, within 8.5 days, we were able to save over 6,000 corals in the Dry Tortugas. It was such a success that we're going to be able to replicate this effort again. And I should point out, this is just in addition to the work already being done by National Park staff. They're working out there every day. We were just able to come in and help with some of the heavy lifting by having a very intervention specific mission. One of the other great things about some of the trials and tribulations we had as a part of the Florida response was being able to share those lessons learned with other parts of the Caribbean. We've had multiple learning exchanges with managers and scientists from all over the region. And I've just pulled just 2 examples of many of some of the work being done in these other regions. You can see that US Virgin Islands has taken on treatments. They've done almost 10,000 corals. Cayman Islands have treated about 14,000 corals in just 6 months. So this is a tool that is being widely used, and that we are really grateful that we were able to do the R&D to help share and save some of that burden. So thinking a little bit into the future, no one's thrilled about putting antibiotics out on the reef. We know that this is a concern. We have calculated out the amount that we put out. It is really very small, particularly compared to other sources of antibiotics going into the ocean. And it is very topical. We make sure that the treatments go directly into the corals as opposed to leaching out into the water. But nevertheless, we want to address any unintended consequences that we might not be seeing. So starting in January, we're going to conduct some efforts on coral, some research on coral physiology and how they might be responding to these treatments. And then there's still a lot of work being done looking at alternatives to antibiotics. Mote Marine Lab, and particularly Dr. Erin Muller, has been working with ocean alchemists in the US Virgin Islands for some alternative non-antibiotic treatment products. The Smithsonian and UNC Wilmington have been working tirelessly for a while on some probiotic solutions to see whether that might halt or prevent reinfection from SCTLD. So there's a lot of work that's still going on in terms of intervention and the future of intervention. All right, so I'm going to put the intervention to the side for a little bit. Again, that was basically saving the corals that are still on the reef that are providing these ecosystem services. And I'm going to shift gears a little bit into this idea of rescue and propagation. So the story of coral rescue and SCTLD goes back to 2015. And my colleague Cindy Lewis and I, Dr. Cindy Lewis of Keys Marine Lab, we were working on the pillar coral project. And we got really tired of seeing this. So almost every pillar coral we would come across would just have tissue flaking off. We knew that within a few weeks, this coral was going to be dead. And Cindy had the really great idea of saying, well, why don't we try to save some of these? Maybe we could bring some of them onshore and just keep them from dying. It was a great idea. And we're really glad that we did that. So we partnered with a bunch of extraordinary collaborators who are listed up here. These are folks who have onshore nurseries, offshore nurseries, and were willing to take these pillar corals into their care and just basically keep them healthy, or in some cases get them healthy. A lot of these, it was the very last fragment of that individual. It was very sick. And so a lot of the development of the cures for SCTLD were developed as a result of this pillar coral project. So over 500 different fragments were brought in. You can see from this map, all of the green dots were corals that we were able to get fragments of. The black dots were ones that died before we were able to get to them. And you can see that within the Florida Keys National Marine Sanctuary, most of those corals, fragments of them were rescued and brought to safe facilities. And from the graph on the right, you can see how important it is that we did that. So of the ones that were brought in, 87% of those are now dead in the wild. Another 12% are either diseased, actively diseased, or nearly extinct in the wild. There's literally one that's still healthy, and that's a result of numerous antibiotic treatments that seem to have held really well, and that coral seems to be doing okay, but we know that it could also go at any day. So this effort is really what saved the Florida pillar coral population, and I can't express enough thanks to these partners for their willingness to take this on. So what does that look like? There's a bunch of steps. The collection is a little bit soul killing. You should never take a hammer and chisel to a coral unless it's a last ditch effort and you have all of the permits in place. But knowing that this was really the last chance for some of these guys, we take fragments off the reef, hand them over to people who are much better at this than I am, who know exactly how to trim them and make them healthy and mount them onto tiles, and then put them into long-term care. And some of these facilities are inside buildings where the water and the temperature and the lighting is very meticulously controlled. Some of these are offshore nurseries, partners like Coral Restoration Foundation and Reef Renewal, who keep these in the water, but away from the reef, and they seem to be pretty safe from stony coral tissue loss disease as a result of that. So that's basically maintaining the genetic diversity and the population of these corals as we know it. But that's not really enough for what we need for the future. We need to be not just having 500 corals, we need to have 5,000 or 50,000 or 500,000 if we have any hope of doing successful restoration. And we need them to be different from each other. And the way to get genetic diversity is really only through sexual reproduction. And that, again, provides genetic diversity, which can confer resilience, not just to SCTLD, but to future climate conditions, future water quality conditions, and really increase those populations exponentially, much more so than just holding a single coral. And so the only way to do this is during spawning events. Corals spawn once a year. It's kind of cool. These fuzzy rocks, like they do something at this for like 10 minutes a year. And it's pretty spectacular. If you ever get a chance to go see a coral spawning event, I would 100% recommend it. It's one of the marvels of nature. So here's some pillar coral that are spawning. The one on the left is a girl. The one on the left is the one on the right is a boy. And so for years, we would go out and try to take advantage of this and help these guys out. These corals are miles apart from each other on a reef. The odds of the eggs from the one over here and the sperm from the one over here running into each other in the middle of the ocean when they're miles apart to make baby pillar coral is essentially zero. So we wanted to help them out. So what that looks like, we had various collection methods, various bags, syringes, and we would station divers on each of these corals, wait for them to spawn, collect those gametes, and then the divers would race back to the boats would race across the ocean, we would put all of these gametes together in buckets and hope that they fertilized and hope for the best. Again, getting them back to facilities which have much more talent than I do at knowing how to provide the right conditions for larval coral and to get them to settle onto substrate and to start to grow. And again, we're really grateful for our partners who took those really important steps on. And so through this, we were able to create probably the first baby pillar coral in decades in the Florida Keys. And that picture on the bottom, right, it's a baby picture, it's adorable. But really, this got scarier and scarier every year because we were losing corals so quickly. We had to keep moving west because SCTLD was also moving west. And eventually we ran out of reef. We ran out of pillar corals that were still alive, and we knew that this was going to be the last year that we could conduct this in-water fertilization. Luckily, we were really able to hand off the baton to our colleagues at the Florida Aquarium, who, using the pillar corals rescued as part of the rescue program, and also some of the knowledge that we had been able to gain over multiple years of field spawning, they tricked these corals into thinking it was the right time to spawn. So they controlled the temperature and the lunar phase and the sunset. And instead of now having a diver miles away from another dive team trying to race these across the reef, they now had 60 individual corals in one tank, all spawning at the same time. And so now, instead of getting maybe 3 to 5 babies each year, they're getting 300 or 3,000 or 30,000 of these babies each year. And so really, again, I can't express how we felt about being able to hand off that baton at the time we knew that our time being able to do this on the reef was really over. So based on some of these successes, and also on the fact that SCTLD was becoming a hugely recognizable problem, NOAA and Florida Fish and Wildlife Conservation Commission, FWC, partnered up to create a rescue program for all SCTLD affected species. This is a huge undertaking. They've collected over 2,000 corals representing 20 different species, and have partnered with a lot of aquariums and zoos around the country, and in particular the AZA, Association of Zoos and Aquariums, to create safe havens for these individuals. A lot of these facilities are also working on spawning, and so the hope is that they'll be able to mimic some of these pillar coral successes and create the thousands and thousands of babies that are going to be necessary from all of these species for future restoration. This is a great story. It's not my story to tell. This has really been a huge effort by NOAA, FWC, and the partners there. So I really encourage you to look more into that and to learn more about that. There's great media and press out there about it. So take a look and go visit some of these corals that are being held at facilities all over the country. So the goal of this, of course, though, is never to just hold thousands of corals in aquariums. That does nothing for the reef. That does nothing for the tourism and the fish and the keeping the water out of my living room during a hurricane. All of that requires rebuilding the reefs, and that's where restoration comes in. And the most ambitious project to date regarding restoration is Mission Iconic Reefs. This is spearheaded by NOAA, but in partnership with many, many different organizations, some of which have been doing restoration for years and years. And so there have been 7 reefs selected throughout the Florida Keys National Marine Sanctuary that are being targeted for restoration. So moving coral cover back to what it historically was. And here's sort of what that looks like. So there's different zones of the reef, and you can see that coral cover, which is currently on a lot of these reefs between about 3 and 5%, is being planned to be restored to 11 to 35%, depending on the zone. And you can see that this includes a lot of the established restoration species, so the staghorn and the elkhorn corals, which have been being outplanted for years, and the science on those is great, and the experience that has been gained really sets those ahead of the game. But also some of these boulder corals and brain corals. And the fragmentation and the outplanting of those is much newer, but is advancing so rapidly, and is going to be a really critical part of restoration, both for iconic reefs and reefs around the world. And then, of course, with this propagation is having the sheer volume of babies, and in particular the genetically diverse babies that can go back out on the reef. So what does the future of Florida's reefs look like? Well, the crystal ball is still a little murky, to be honest. We have great tools in our toolbox. We can save these SCTLD affected corals. We can save the ones that are still out there that are contributing to the existing coral cover and providing those ecosystem services. And the tools for propagation and rescue have advanced unbelievably within just the last couple of years. So yeah, rescue and restoration, fragmenting, getting corals back out on the reef. We have these tools, we're learning more about them every day. And so we have what we need to be able to do successful restoration and maintain coral reefs. However, we still have a downward trajectory. part of it is SCTLD. There's so much that we need to learn, and we're grateful for all of the work being done by partners all over the country to help answer some of these basic SCTLD questions. And that's going to help us learn how to slow spread, or how to do better intervention treatments, or how to make sure that restored corals don't succumb to SCTLD. But then, of course, all of this, going back to death by 1,000 cuts, we need to continue to address all of the other cuts. Environmental conditions for corals on the reef are not awesome. We need to address everything from local conditions that might look like sunscreen and marine debris, global conditions that look like water quality and climate change. So really the future of Florida's reefs is up to each and every one of us. So I'd like to finish here. Thank you guys for your time. Thanks again to Reef Relief, and open this up for any questions.
All right, thank you so much, Dr. Neely. That was an awesome presentation. I hope everyone learned something, because I definitely did. We do have a few questions. We have a few minutes to answer them. So the first question, it's a two-part, and it's from Sebastian. It's, how does stony coral tissue loss disease affect corals? For example, does it interrupt the symbiosis that's happening? And then his follow-up question to that is, are there seasonal patterns that you're seeing with stony coral tissue loss disease?
Those are great questions. There are a lot of people actively looking into the physiology of SCTLD. I would encourage you to check out a paper by Jan Landsberg, which I believe was 2020, the end of 2020, who really was able to get in and look at the histology. And what it does appear, it definitely appears that this might actually be more of a symbiont disease or zooxanthellae disease than a coral disease, that a lot of the breakdown of the coral tissue seems to occur near the basal layer and around the zooxanthellae. So there's a lot to be learned, but people are really interested in that symbiosis question. The second part of that about seasonality is also a spectacular question. Most diseases, most coral diseases, are worse when corals are stressed. And in particular, that happens in late summer when water temperatures are at their warmest. That does not seem to be the case with stony coral tissue loss disease. In fact, bleaching or paling, which is very stressful to corals, seems to correlate with a decline in SCTLD, which is another suggestion that this might be a zooxanthellae disease. Because when the zooxanthellae disappear from the coral, the disease seems to become a lot milder or disappear entirely. But that's really the only temperature-related correlate that we've seen. It doesn't go away in cold weather. And yeah, as long as a coral doesn't bleach, there doesn't seem to be a seasonal component. Great questions.
Awesome, thank you. And then we have a question from Sarah, who wants to know, what proportion of your treated corals have survived long-term?
So we only have data up to, let's see, in January, we'll have a handful of corals that we've been working with for 3 years. The vast majority of our analyses have been done on 2-year treated corals, and our mortality rate there is only 4%. So 96% of them are still around.
All right, and then we have a question from Kathy. What can we do to get the word out to the general public about what's going on with our coral reefs? And I want to piggyback on that and also adding, what are some things that our attendees can do to help our corals with stony coral tissue loss disease?
Yeah, thanks. I mean, we really rely on you to get the word out. Scientists are notoriously horrible communicators, so we're really grateful for organizations like Reef Relief and all of the people that are good communicators, including the general public who shares this information with their neighbors and their families. So yeah, keep doing that. There is information available on the Florida Keys National Marine Sanctuary webpage, DEP webpage. FWC has some great info on there. So there's information out there, but we need people to share it. In terms of what individuals can do, so there's not a ton that can be done about SCTLD. In other places, in other parts of the Caribbean, there has been some authorization to allow trained folks to conduct interventions on corals. We're not there here in Florida, but maybe someday, so keep an ear out for that. But we do really need people to be thinking about all of those other cuts and how can you make corals' lives less stressful, and that again, includes everything we know that you need to do about climate change and water quality. And all of those little, very local things, being careful about where you anchor and your sunscreen use and your marine debris. So all of those little things add up and really do make a difference in the health of coral reefs.
All right, great, thank you. And then our last question, just really quickly, is from our board member, Bob. So Bob, if you want to go ahead, ask your final question.
Yes, Karen, is there any evidence that the treated corals, the treated boulder corals that appear to be cured are growing back into the dead areas?
There is, although it's not rapid, but corals without SCTLD don't grow very rapidly either. Conditions aren't great for reef growth out there right now, but we do definitely see some regrowth, particularly in areas where growth was good before SCTLD. There's reef areas where corals do better than others, and correspondingly, the growth is better on those than others.
Thank you.
Awesome, thank you so much. All right, I'm going to turn it back over to Tricia, our president, to introduce our next speaker.
Great, thanks everybody.
Thank you so much, Dr. Neely, for your great presentation and your amazing preservation and restoration work. Thank you. Our next presenter tonight is going to be Walt Stearns. He is a highly credible photojournalist whose work spans over 35 years. He's featured in several publications worldwide in the field of boating, sport fishing, sport diving, and marine natural history. Walt's photographic work are perhaps most recognized in feature articles published in magazines like Discover Diving, Ocean Realm, Scuba Diving, Sport Diver, Skin Diver, to his current position with Scuba Diver's US publication, Destinations. Walt's editorial, photographic, and design skills are also applied to the creation of web and blog sites, full-page ads for print, online publications, and brochures for clients like Wakatobi Dive Resorts, Caradonna Dive Adventures, and the Palm Beach County Diving Association. In addition to his editorial work, Walt's expertise in marine wildlife and closed-circuit rebreather diving has been called upon in numerous marine-related documentary film projects with BBC, National Geographic, and Discovery Channel that have aired internationally. His presentation for us tonight is Goliath Grouper Conservation from the Eyes of a Photojournalist.
All right, thank you for introducing me. Everybody hear me okay? All right.
Yes.
Yes.
I'm going to bring up the presentation here and get everything going. Everybody see that good?
Yes, looks good.
Okay. All right. So what I'm going to talk about is probably a fish that has kind of become very near and dear to me as a diver and resident of Florida, the Goliath grouper, the Atlantic Goliath grouper to be specific. And I'm going to portray this in the photos that I've taken of this fish through the years, as far as what the fish has gone through and what a spectacular animal is, because it's, after all, the largest bony reef fish of the tropics worldwide. And there's generally 3 species of Goliath grouper, but tonight I'm going to talk about the Atlantic Goliath grouper, and so we're going to move on to that. And these fish are very special fish. They're quite large, very charismatic, and if you're in Florida, you're very likely to see them if you're diving, particularly in the reefs in the East Coast around Palm Beach County up to Stewart, as well as in the Southern Gulf of Mexico on some of the offshore wrecks, and occasionally in the Florida Keys on some of the wrecks and reefs down there. Now, best way to identify the fish is, excuse me. They have, generally come in 4 different color phases, which they can change from one to the other whenever they feel like it. First one in the upper left is what's known as the classic color phase, which has that tan and beige, a little bit of gray coloration to them, has that marbled look. The dark phase, which of course the body color is much darker overall, but still the pattern is still pretty much present. Then they have the light, or what I call the blonde phase, where they can go really light color down almost down to where the only thing that really stands out is the tiny black spots that they all have, and they're like fingerprints. You can tell one fish from another just by the presence and position of those spots. And then the last one I call the monochromic color phase, where the body is more uniform from head to toe as far as following one color, where the mottled darker brown or gray is almost totally non-present. And this coloration is a little bit rare. You don't see it as often, but they do display that. Now, probably one of the things I want to get into is also, the question I always get asked is, how big do these fish really get? Because a lot of the textbooks will talk about them. You go on the web, do a web search, Wikipedia, and they'll throw out these arbitrary numbers of 800 to 1,000 pounds. And the reality is the fish rarely get past 500 pounds. I mean, a 550-pound Goliath is really big. The largest one ever landed by the IGFA record books, and that was back in 1961, weighed 680 pounds, and it was taken off of Fernandina Beach in North Florida. Picture I have next to it is an animal that was caught in Brazil, which they claimed was 800 pounds, or excuse me, 900 pounds, but there was no confirmation of that weight. It was just speculative. But looking at the pictures, comparing the two, the weight ratio of that fish was probably in the same category, between probably 650, maybe 700 pounds at best. In both cases, you're looking at a fish that's generally 8 feet long. But the average Goliath grouper that you see on a lot of the wrecks and reefs are generally going to be in the, as far as the big ones, 300 to 450 pounds, occasionally over 500. Now, the question too is, where would I meet one? And these fish are generally, they're not a deep water fish, surprisingly enough. I mean, they will be found deeper than 150 feet, but quite often you can find them as shallow as 10 feet of water underneath a dock. The range, formerly was throughout the entire coast of Florida, all the way around through Texas, down along the Central American coastline, all the way down to Brazil, and also in parts of Western Africa. Now, the most common place where you see young Goliaths would be on reefs, but most often they prefer wrecks. And one of the questions I've had people ask, why a wreck over a reef? And kind of a good analogy is, if you were given a choice, which would you rather live in, a mansion or a tent? And in comparison, say, nice big wreck is like living in a mansion. So it provides a very good home for these very big fish. And they are pretty much a homebody. The Goliath that you see on a wreck 1 year will likely be the same fish you're going to see next year, and the year after, and the year after, and the year after, until the day it dies. I mean, and that can be a mighty long time, because these are very long-lived fish that can live 35 years or more. And they can be either loners, or they can form up colonies, often on some reefs. My diving ventures in Palm Beach County, I can go drift down a section of a reef where I can go 100 yards or more, not see a single Goliath, and all of a sudden hit one spot, and voila, there's 3, 4, 5, 6, 7 Goliaths in one particular spot on the reef, and they tend to stay there through most of the year. And as a group, surprisingly enough, they have their pecking order, but they seem to be a little bit more sociable than some of the other species of groupers that are found on the reefs. Now, when you look at fish as big, you kind of wonder, it's like, what kind of threats do they have? The adult fish, not very many. Once the fish gets past 150 pounds, it's pretty formidable, it's big, it's strong, and most other predators don't want to mess with it, but occasionally, one can get attacked by a shark, sometimes killed by it. This one here was obviously attacked by a shark. You can see the bite scar in its back. And looking at the size of the bite and the way the bite was given, a likely culprit would have been a tiger shark, which I have pictured in the lower right-hand corner. And that's one of the larger varieties of sharks. Bull sharks can also provide some threat to them. Another variety of shark that, surprisingly enough, poses no threat to the Goliath groupers, and quite often they will be seen together as a lemon shark. And I really like this photo here that I have up here, because to me it's a very special photo, because in addition to being involved in keeping Goliath groupers protected in state waters, I was also involved in getting the lemon shark protected in Florida state waters, and having the two together in this portrait was kind of like to me, a big landmark for my efforts. Now, outside of natural causes, I mean, number one threat to the adult fish is man. We're enemy number one. And in the past, it was commonly spearfishing that posed a big threat, or at least the most visual. You can see this one here is pretty much on his way out. He was mortally wounded, shot in the head by somebody, and just left there, which is kind of a sad state of affairs. But probably the bigger threat currently, because the fish is now protected, has been since 1990, so you can't spear them anymore. But the law doesn't really protect them against hook and line fishing, especially by recreational fishermen. And lately, more and more recreational fishermen are targeting the fish because they like the idea of fighting a big monster and bringing it up. And you can see in the picture here, this fish has a pretty good sized hook still stuck in its jaw, where they cut the line off but left the hook. They didn't bother to take it out, and fish managed to make it back down to the reef and survived. During some of the studies that FSU was doing, when they were catching Goliath groupers to take tissue samples and everything, oftentimes they would pull a lot of fishing gear out of the throats and mouths of these fish, where they've actually not just being directly fished at by fishermen, but also as a bycatch, because the fishermen were targeting other fish. And like a big predator will do, when they see any kind of fish fighting on the end of a line, it's compromised, and therefore makes it easier target to be consumed. And in the process, the Goliath not only may steal that fish, but he ends up stealing a good portion of the fishing tackle, the hook, sinkers, and line all together in a nice big ball like this one here. But the other damage that can happen to these fish, especially on the deeper wrecks where the larger fish live, 90 feet down below to 150 feet, is when they're brought up rapidly from that depth, Goliath groupers can actually experience something very similar to the bends. It's often referred to as hyperbaric trauma. It's where the gases in the swim bladder get pushed into other parts of the body and collect in common areas in the stomach, as well as the eyes of the fish. And when the fish hits the surface, it gets that pop-eye look to it. And that can cause a lot of damage to those eyes if the fish isn't properly vented before release. And if the fish is brought up from too deep, the damage is too severe for the fish to survive, and it dies. But in some cases, they do make it back to the bottom. And you can see in this shot, we have a Goliath with a eye that's been damaged through gas expansion and has now become infected, and is pretty much going to lose its eye and end up like this one here, which has lost one of its eyes. It still has its left eye, and because with that, it still has a chance of survival. If it were to lose both eyes, the fish would starve to death and eventually die. Now, one of the other things that's kind of become a new threat to this fish is mercury, methylmercury. And mercury is found in a lot of the intercoastal waterways. It's found in pretty much all fish in small, minute amounts. And generally, the longer fish live, the more mercury will accumulate. And the mercury gets into the Goliath groupers, and they, as they get older and bigger, they can cause neurological and muscular deformities and basically start to cripple the fish. In this case, this one's pretty much his spine is curved. He's not able to move his tail as efficiently, so he's more susceptible to getting attacked by a shark or somebody else, and also become less apt at being able to catch his own prey. This one was on a wreck off of Boynton Beach. He was nicknamed Quasimodo because of the hump. Now, the mercury levels too in these fish have been studied by FSU, and what they're finding is a disturbing trait in these fish, that the level of mercury is higher than pretty much any other fish caught and consumed, higher than swordfish, higher than sharks, higher than a lot of other fish. I mean, to the point where, you know, once the fish gets into that 300-pound range, the mercury is way in the danger zone of what would be allowed to be consumed. And on the chart here at the lower area, you see 0.5 parts per million. That's considered the danger zone by the FDA, the CDC, and as well as several other agencies as far as what is deemed safe and unsafe to consume. And looking at the chart here and seeing how high up it goes, it pretty much makes the Goliath grouper probably the most heavily mercury contaminated fish on the planet. Now, at one time, these fish were extremely plentiful, as I showed in the map, you know, the range. And during the 50s and 60s, you know, the fish were targeted at times, you know, just for trophies to hang on the fish dock, the draw up business, and because they were so readily caught and people, you know, took them in great numbers. Then they were started into being more targeted by commercial fishing, and that really picked up in the late 70s, early 80s. And as you can see on the commercial catch note, at 1950 was over 97,000 pounds annually. By 1977, it had dropped down to went up to 126,000, and then 1998, 148,000 plus, and then the fish almost dropped to zero before 1990. At the same time, they managed to close the fish down to commercial fishing. They set a state and federal moratorium on them, so no one was allowed to fish them anymore. But by that point, the numbers of these fish were beaten down so bad that they became very rare. And off the East Coast, where I live in Palm Beach, during that time period, I could have counted the total number of Goliath groupers from as far south as Fort Lauderdale to the upper end of Palm Beach County, Jupiter, on one hand. And now the population has rebounded enough to where we see them more regularly in that area. So probably, you know, where I'm going to jump to is, you know, kind of disproving some of the myths about these fish. And one of the common one I hear out of people who don't know anything about these fish or fish biology in general is, you know, these fish, you know, a 300-pound Goliath or 500-pound Goliath eats 100 pounds of food, you know, other fish a day, which is pretty much untrue. I mean, these are cold-blooded animals with a very slow metabolism, much slower than people. And when you put it in that perspective, my Uncle John weighed 100 280 pounds. And even on his best day, he would be lucky if he can eat 20 pounds food in one day. And so the notion that these fish are eating 100 pounds is pretty silly. Now, what they have found in all the data collections, of fish that were caught, stomach contents evaluated, is that the majority of these fish feed on crabs. And I have a picture of what's called a shame-faced crab or callop crab. They're very common in the Gulf and east coast of Florida. And these fish apparently really love the hell out of these things. They eat them quite regularly. Shrimp are another part of their diet. And then the rest breaks down into a wide variety of fish, mostly small grunts, pinfish, what most people would call trash fish. And on the scale, the species of fish such as snappers and groupers are extremely on the small end of the scale. And generally that's the ones that have been found is because a fisherman had caught one and was fighting it on the end of the line. Goliath sees the grouper or snapper struggling and goes, "Hey, I got a free meal." Boom, gone. And fisherman gets mad at him and starts calling the fish a nuisance. Another belief is the Goliath groupers eat all the lobster. Well, we're the biggest source of eating lobster, and a lot of times I'll look under a ledge and I'll find lobster living under the same ledge as the Goliath grouper. And generally a Goliath doesn't go after a lobster unless it goes out in the open. And even then it's not always a guarantee. Other varieties of fish, small groupers and other stuff will share the ledges and reefs with these fish. There's two shots here. The lower left, there's a large Cabrera snapper, and the lower right is a shot of two Goliaths sharing their ledge with a school of grunts as well as a small grouper of another species. Now, one of the hopes that we all had was Goliath groupers would eat lionfish. So far, nobody's witnessed that. I have a shot here of a large Goliath where I literally tried to get to eat this lionfish. I kept flushing it out from part of the wreck, pushing it out in the open. The Goliath would run up, take a look at it, then go back away. The lionfish go back under the ledge. I'd flush it. I did this 4 times, and the Goliath was a no-taker on that situation. So he just let it go, and I didn't get to see something that we all hope would happen. Now, spawning. These fish have a very unique spawning behavior that pretty much is what I call a marathon romance. It's over 2 months in length, but they will migrate from long distances and accumulate on various wrecks and reefs on the southeast coast of the US and the southern Gulf of Mexico. And they'll spend up to 2 months in that location during this long courtship. And they'll gather in large numbers of up to 40 to 60, as high as 90 plus fish. And they'll travel from large number of distances. Now, in Palm Beach, I'm going to keep coming back to Palm Beach because this is where I live and this is where I really follow these fish closely. Altogether, we have only 6 spawning aggregation sites these fish migrate to, and have the list there with the depths of these sites. And the deepest one is a hole in the wall, and that's 140 feet of water. And that was the first spawning aggregation site to emerge on Florida's east coast in over 3 decades. And the first aggregation took place in August of 2001. And the total number of fish that showed up that year was 27 fish, which was for us a huge number because we went from 0 for 3 decades to now 27 fish. Over the next couple of years, the number of the fish grew. So the next site that became a new spawning aggregation site was a broken up barge and a bunch of pilings from a high school that were dumped to create an artificial reef called the MG-111 Warriors Reef. And that's in 65 feet of water, also off of Jupiter. And these fish gather around it. And the picture I have above the list of sites is taken on Warriors Reef. And you can see a group of these fish here gathered there during August and September. And these fish were, they started coming there about 2003. After 2003, the number of fish showing up grew. So the next spawning aggregation that emerged was the Zion Train Bonaire Wreck Group, also in Jupiter with 90 feet of water. And that became site number 3. And the same year, a wreck down in Boynton Beach called the Castor Wreck, which is a great big freighter that was sunk as an artificial reef off of Boynton Beach in 113 feet of water, became site number 4. And then a couple of years after that, the Anna Cecilia and Mitzball became site number 5, followed by Gary's Gray, which is a reef site up in southern Hope, Martin County. And that site is in 80 feet of water. And these are our 6 sites. Now, this is backing up. This is the hole in the wall. This is back when the fish first started showing up in the early 2000 period. MG-111, Zion Train. In that shot, there's 65 fish total. I sat down and counted them all. The Castor and the the Mitzvah and Cecilia. And when you really think about the number of sites these fish are, this is the Palm Beach coastline broken down. You can see the MG-111 Zion Train at the very top. North of that is Gary's Gray, hole in the wall, Anna Cecilia, and then on the second map is the Castor Wreck. Now, putting these two LiDAR images together end to end, you're looking at a coastline of 65 miles. And when you look at it total, that's not a big area where these fish are. So to have the assumption that these fish are everywhere is kind of a misleading too on that. So what happens when these fish gather? The courtship is kind of a bit of a mystery. We do know where they go. We know where they come from. We know how long they're there. And we know that the fish spawn at night. Now, their closest kin that spawn at night is Nassau grouper. And Nassau groupers spawn during the full moon, usually around December or January, versus the glass grouper spawn in August, September, late summer. Now, early on, for a lot of years, there was a lot of conjecture that the glass groupers also spawned on the full moon, and it was later discovered that the fish actually spawn on the new moon, the dark of the moon. So that kind of presents a problem for actually witnessing how these fish spawn, because it's on the darkest night of the year, of the month, and it's happens they know when it happens, which is generally between the hours of 10 p.m. and 4 a.m. So when you think about, okay, how dark is that? Put yourself in a closet, shut the door and turn the light off. That's how dark. So to be able to see what's going on is quite difficult. I've tried to do it using different kinds of lights and found that these fish are highly responsive to lights to the point where they panic. Now, like all groupers, they're broadcast spawners, and they swim together either as a group or in pairs, move up into the water column and release the sperm eggs into the water and they get fertilized and drift away. Now, during the day, when these fish are collected together, they'll go through a wide variety of those color schemes. And each one is different, just like a fingerprint. Everyone has its own signature. As you can see here, I call the variety pack. You can see the dark phase, the classic phase, two blonde phases, a semi-blonde phase with a dark patch on one side of his head. But seeing one fish in a light phase and the dark phase would think, okay, one's male, one's a female. Well, not exactly. I've seen fish kind of interact together where they're both sharing the same color scheme, like these two in the light or blonde phase. They can also take on a kind of an interesting color pattern too, where they can go by color. In the larger image, you got this one here where his head is almost entirely cream color on both sides, or they can take on, like the one in the upper left-hand corner, is actually presenting a bicolor where he's literally split the color in half, where one side is light, the other side is dark. And this fish in particular has got a name. He's very well known to us in Palm Beach. His name is Braveheart. And he was given that name for that dark coloration on one side of his face, after Mel Gibson, the scene in the movie Braveheart, where Mel Gibson put the blue paint on one side of his face to make himself more fearsome. Now, an interesting thing kind of happens during the spawning season, as it gets closer to that new moon phase, a large number of scad scarp minnows will suddenly appear out of nowhere and start congregating around these Goliath grouper and start following like mobile structures. Wherever the Goliath grouper goes, they go. And kind of the adage, and to the point where some of these schools will get so big, they can hide as many as 5, 6 Goliath groupers at one time. You'd see this big ball of bait, and you're wondering what are they crammed around, and you swim to it and get into the ball, and you realize there's a Goliath grouper looking back at you like this. Now, kind of one of the best known analogies is big fish eat small fish. Well, in kind of this case, what's happening is it's kind of a little bit of a reversal here. These little fish are going to eat big fish, not like piranhas per se. What happens is what they're waiting for is the Goliath groupers to spawn. As I described earlier, these are broadcast spawners. They go up in the water column, release the sperm and eggs at the same time. And when they do that, it creates a big buffet table for these small fish, and that's what they're waiting for. They're going to feed on those eggs and, get a nice full meal if they're lucky. But at the same time, they also have to evade not getting eaten by other predators like these jacks, shot. Now, if the eggs manage not to get eaten right away, they drift off. If they get fertilized, they'll hatch out and create, you got now a Goliath grouper larvae. And this diagram is what the fish looks like when they first hatch. The picture here next to it is a larval grouper that I photographed out in the Gulf Stream at night during what's called a black water dive, where you're drifting in the Gulf Stream, going with the current, looking for very small creatures. And this one is quite small, probably overall measures just around 1/4 of an inch in length. Now, if he's lucky, he or she is lucky, and doesn't get eaten by something else during that journey, they'll eventually settle into the intercoastal waterways and begin their juvenile life in the mangroves and seagrass beds all around the Florida coastline. And this is an essential environment for these fish because they will spend the next 3 years of their life in this environment, feeding on tiny shrimps and other things, and until they get to around 3 feet in length and 3 to 4 years of age before they journey out. This is a angler here is holding a young 2-footer that was taken out of the mangroves in Florida Bay. And now when these fish are in this environment, they're still not free and clear. I mean, they're very susceptible to sudden temperature changes. As witnessed back in 2010, we had a severe cold snap that pretty much decimated the juvenile Goliath groupers in the intercoastal waterways from Tampa all the way down to Florida Bay, Biscayne Bay. And that one cold snap pretty much wiped out 3 years of juvenile development. So the clock had to be restarted. Now, other threats to the fish when they're in the intercoastal is they are very susceptible to sudden changes in salinity. So if there's a lot of rain and the water becomes less brackish, becomes more fresh, it poses a threat and can kill the young fish. If the water gets too contaminated from pollutants, again, it'll kill the fish. And just our tendency for overdevelopment, where we're tearing out mangroves and putting in seawalls, is removing that essential environment for these young fish to grow up in. So they face a lot of perils when they get during development. Now, where I'm coming from with a lot of this information is a huge amount of data has been collected on these fish. So another argument is, well, there isn't very much data. We don't know anything about these fish. Actually, there's probably more research published on this species grouper than any other species grouper in the world. And the leading researchers was FSU's University of South Florida, or Florida's South University, doing the studies when these fish, their studies began when these fish were first put in protection in 1990, and it's still being carried on today for the most part. And they've put together a huge amount of information about the whole natural history of how the fish develop, where the fish live, the site fidelity, where these fish move back and forth, how long they live in places. And also by catching the adult fish, they've been able to explore, they take tissue samples, they cut away fin rays like this here to determine the age of the fish. In the process, they also collect DNA samples, determine the sex of the fish, and it all goes into a database. So they have a pretty good understanding what fish are out there, how long they live, what their threats are, what they're eating, checking the stomach contents. Here we got 2 fish, one that hasn't been caught and one that has, where he's had part of the fin rays taken out. He's also bearing a bright tag, and likely he was also had a small receiver transceiver implanted underneath the tissue near the stomach cavity that will be picked up by a piece of equipment called a VR2 or Vemco Marine. And you can see one here strapped to this post. It's actually not the big tube, but the small black one at the very top. Now, a fish with these transmitters, anytime they pass within 100 yards of it, gets recorded, picked up, and logged into the database. And it'll keep picking up fish up as long as the fish is there. And a lot of times they'll put these monitors on the aggregation site to monitor the number of fish coming here. Now, the cylinder below it is actually a passive hydrophone to record the sounds these fish make, because during the day and night they make a variety of grunts and booms to warn off intruders, communicate to one another. They're still trying to understand what a lot of those sounds mean. So that's probably the newest form of research that's going on these fish. But they have found by using these transmitters, because other researchers use them. And what's part of what's called a fact array, where the number of VR2s number in the thousands on our coastline, from the very tip of Florida down to the dry Tortugas, all the way up to Maine and back around again along the Texas coastline. So they can share the data points whenever a fish passes. So if someone's studying goliath groupers, they can go into the fact array data system and find their fish if they happen to pass other VR2 receivers outside of their own receivers for their own research. And during the study, they found that some of these fish are migrating, making a journey of over 348 miles to go to one spawning aggregation, and can do it as soon, travel time can take anywhere from a few days to 2 weeks. They take their time to get there. So like I said, it's a marathon romance for these fish to accumulate. Now, the big thing that a lot of people love to do is they love to dive with these fish. They're highly charismatic megafauna. They're big, they're inquisitive, and but really no threat to you. I mean, they will come up to you. Sometimes if they don't like you, they'll make a booming sound. It'll either be a one loud boom or 3 consecutive booms, and as a way of saying go away. If you fail to follow that advice, they might make you might get a fish that might make a false charge at you. If that doesn't work, they oftentimes just run away from you and go sit under a ledge and sulk because you're not running away from them. And they don't understand why. But for the most part, a lot of these fish are, especially in the Palm Beach area, are quite friendly and tend to be quite big hams to underwater photographers, to where it's kind of like, make sure you get my good side, as I like to call that here. And even with posing with your fellow divers, this one was done as a kind of a request. I had a request, we need a person next to a goliath grouper wearing a pair of white sunglasses. For what reason, I have never found out, but I shot it anyway. And again, this was a wood fish that we very well know, Braveheart. You can see him here in a more normal color pattern, unlike his spawning color where he has the one side dark, one side light. This is him now. And but for the most part, they're just big passive babies. So they're quite an enjoyable fish to visit. This parting shot here is someone took of me, basically what I call swimming with my buds, and kind of shows you how big the fish are and how easygoing they are. So that pretty much ends it. So, if you have a question, feel free to ask.
Awesome. Thank you so much. Those were beautiful pictures and really great information. We have a question from Gabriella. She would like to know, what is the current conservation status of the Goliath grouper? As she heard recently, as many of us have, that they are considering reopening the fishery as well.
Yeah, sad to state, the FWC, there's been a contingent of people been pushing to have the fishery reopen since 2005. And we've been pounding on the FWC with the data showing all the reasons why the fish can't be reopened. But last year, they made the push to try to explore the possibility of reopening the fish to a limited harvest. And this year, we had another meeting on the topic, and they're still going forward with it. It's not absolute, but it's disturbing, but there are some good points that we kind of take as a win. One of which, if we hold their feet to the fire, is they propose that a limited harvest would be open to only 200 fish based on the lottery system. And that lottery would be quite expensive, in the tune of $500 to go kill one Goliath grouper. Now, in order to get on that list, you got to pay a $10 entrance fee to get into that lottery. And if your name comes up, you have to pay it for that kill tag. And you can't give the kill tag to somebody else. You have to use it or not use it. And the other stipulation on that too, again, if they hold to what they said they're looking at doing, is the size of fish you're going to target is going to be between 2 and 3 feet in length. That pretty much makes it limited to only the juvenile fish that are found in the intercoastal waterways, not the big adult fish like I showed here in a lot of the photos. Now, my only question is who's willing to pay $500 or let alone $100 to kill a 3-foot fish? To me, it's quite expensive. And also, you know, the bigger the fish is, the less likely you're really going to want to eat it because of the levels of mercury in the fish. The other thing that was also proposed that they would make exclusion zones where this limited harvest could not take place. And what they put down was Palm Beach County, Broward County, Dade County as a no harvest zone. That includes the intercoastal. And we jumped back and proposed that, well, if you're going to do that, we'd like to see Martin County included, because really the population of Goliath groupers on Florida's East Coast are really in Palm Beach and Martin County versus Broward and Dade County. Broward County, the Fort Lauderdale area, there's a lot of wrecks off the coast, but very, very few fish in that area. It's a long answer, but that's where the status is currently.
Right, so our next question, you've been, with these Goliath groupers for a very long time and diving with them. Can you speak a little bit about what you've seen in terms of the changes in populations of where you've been diving for years back then and versus now? How has the population changed? Has it gotten bigger or smaller?
Well, I started diving in the early 70s, mostly in the Florida Keys. And through that period, I actually never saw a Goliath grouper on any of the reefs during that time period. It wasn't until the late 70s I would run into one or two on a wreck or a reef. Really, the place I started seeing them was actually in the southern Gulf of Mexico on trips out to the wrecks way offshore, and that was in the late 70s and 80s. And at the same time, I was also diving Palm Beach County, Fort Lauderdale, and Broward County and Palm Beach County. And in Palm Beach County, in that time period, I didn't see any up there until my first one I saw up there in Boynton Beach was actually in 1985. And that was the only one I saw there. And up until about 95 or 96, I started seeing a few more in the Palm Beach, county waters, mostly on the wrecks. And again, the numbers, were very small in number, and we didn't see the first spawning aggregation until 2001. Now, since then, the numbers of fish showing up at these spawning aggregation sites on the East Coast hit a peak of roughly, I would say, between 350 and 360 fish total between those 6 sites in 2012, 2013. After 2014, we're seeing the numbers go down. And the last count, last September, August, September, our last spawning season for this year, the total number of fish was actually in the neighborhood of 150 fish. So they've really drastically dropped in numbers. And that's a very disturbing trend. And a large part of that we suspect is mortality from recreational fishing and commercial bycatch, because we're finding a lot of big hooks left in these fish, damage done from fishing lines, stars very present on a lot of fish. Some of the fish pictures I showed there with the eyes closed out. That was taken this year, and I saw an awful lot of fish that way. It was very heartbreaking to see that.
So our next question is from our executive director, Mill. He wanted to know the monetary value of a living Goliath grouper versus if people were to take them. You know, does the proposed lottery amount equate to what they're worth remaining living?
Well, that's a real good one. The FWC broke down the reason the cost they proposed being so high was really more to cover the cost of management to manage the fishery, make sure they didn't take more than that quoted allotment. Beyond that, it's not making money for the state. It's not, and as far as for the fisherman who catches it, it makes an awful expensive grouper sandwich when you break it down to if you were to fillet one out. You know, when you're talking about a fish that's less than 3 feet long. Probably the only person that might be making money off that really would be the recreational fishing guides, charter boat captains, but they're already doing catch and release as it is right now, so it doesn't change the equation for them. Now, the fish alive, that can be easily drawn up to, yes, a live Goliath grouper is worth thousands of dollars because as long as it's there year after year after year, it's going to be an attraction for tourism and not, you know, economy to not only the dive charter boats, but dive shops that sell equipment, instruction, hotels, restaurants, even airlines, car rentals, because we get people from halfway around the world that come here just to see these fish, especially during the spawning aggregation season of August and September. So, you know, the revenue as projected by NOAA in one of their reef usage studies was in the neighborhood of $2.6 million just for Palm Beach County alone for the economy. So do these fish contribute to that? I would say so.
Awesome, thank you for that. And we have a question from Anne, who is curious with the estimates of removing 200 Goliath groupers. Are we also accounting for, or do we have the data on all the Goliaths that are currently dying from barometric trauma, just from our recreational fisheries?
Yeah, the data is available. That's being collected right now by a couple of biologists. Which is usually the catch shares and what they call a discard list that's reported by commercial fishermen. Recreational, not so much. They'll just simply say, well, we released it. The problem there is if the fish isn't properly handled, it's full of gas from being brought too deep and it drifts off into the horizon and half the time dies from that trauma. So I mean, the number's quite high, but I have not seen what that data is. It hasn't been shared with me yet. I'm waiting for it for myself to see it.
All right, thank you. So I want to turn any more questions over to our board of directors or anyone else that has any last questions. If anyone from the board has a question, just go ahead and you can raise your hand or just queue on in. All right. Well,
I don't have a question, but I just wanted to thank you for the presentation. It was very enlightening and very informative. I appreciate all the work you've done.
Thank you.
all right. Well, thank you again so much for taking the time to present for us. We really enjoyed it. I'm going to turn it back over to Tricia and Mill.
Yeah, well, thank you so much for that great presentation and the beautiful pictures. Mill, did you have anything else to add this evening?
I just want to thank Dr. Neely and Walt for making the time to present those things. Excellent presentations, some of the best I've seen. And I want to thank all of the attendees and members.
Yeah, on behalf of the board, we'd like to thank the speakers tonight and our staff for the amazing work that you guys do. And thank you to everybody for joining, and hopefully you've been inspired to continue to be involved in all of our important work of preserving the coral reefs. Thank you all so much, and good night.
Yep, good night. Thanks, Tricia.
Thanks, Mel.
Good night and thank you.